illustrious members from the pharma industry, starting with Dr. Ranjana, Dr. Manoranjan, Prashanth, and Surya. Of course, we have worked together, many of us, and we're talking about the future, the laboratories, the efficiencies, and here we are to talk about what are the industry's current practices, what are they going to do, and the most common thing that we face nowadays is the failures that you see in the laboratories and the plant, and is there a way to really work on those errors that we encounter. So Dr. Ranjana, when we were talking a couple of days back, she mentioned that it's always easy to talk about the latest jargons on AI and ML and all those things. So we have been hearing everything. So it's nice to hear, but difficult to implement, or at least difficult to understand at this moment of time. So we'll try to see what are the practical aspects which can be taken up in the current scenario, so that people who are sitting in this audience will definitely get something to take back to their workplace and see if you can implement in your areas of work. So to start with, the first question I'll ask to all of you, the panel members, is how important is it to look at new technologies in analysis, and specifically on automation of the conventional analysis that has been done? So one hand, because we have our work is all restricted with the filings, both in the manufacturing and quality and the testing, your hands are tied up of trying to come up with something new. But is there something that you can add a layer of automation? Is there something that you can improvise your way of working? And the next question is, are there any other technologies which can come up in the near future which can definitely take up the work which we have been doing currently in a different manner? So let me start with Dr. Ranjana for this question, please. Yeah.
Hello. So thank you, Dr. Rajiv. Thank you, everybody, for sitting here and giving us your ear, because if you weren't here, whatever we have to say would be absolutely useless. And I will begin with saying that, yes, automation. Can you guys hear me? It's like resounding. So automation is the need of the hour. And we must try to see what can we do with our existing methods. So as Rajiv said, is filed in one method. But that doesn't mean you live with that method. Today, it is evident. We see this entire hall with so many exhibitors, is there are alternate methods. And I think the key for us is, how do I make it mistake-proof? How do I ensure there are no mistakes in the lab? How do I make it easy for the chemists? So there's some chemist delight, because our chemists are overworked. And if there's any problem, what do we hear? It must be in the lab. There's always a problem in the lab, nowhere else. So the more we automate, the better it will get for all of us. But then that's a whole big story, and I hope as the discussion progresses, we will get into some of the nitty-gritties of what are the benefits of automation and what are the downsides of automation, because there's both sides of it. So I say yes, don't stay with the conventional methods if you can. There are many technologies. I have my gurus here. There's NIR, there's Raman, there's so many technologies, even in micro, that we can adopt today. And the FDA is extremely open to bringing new technologies. They want us to. They don't want us to be archaic. It's 2023. So there's like, head up. Industry, get up, wake up, don't do things the same way. And we'll get into it a little bit more.
Thanks. So Dr. Manoranjan, your thoughts on this?
Certainly, if you consider that simplification, there are lots of technique. Simple example is when you perform the analysis on HPLC. There is a run time to separate that majority of the impurity. If you take some one product where that 26 to 30 impurities are there, run time is 160 minutes, 120 minutes. So to improve the productivity of the quality control, it is very essential to look for that option, alternate option like UPLC. So can you reduce to 1/4 run time, which will be helpful for the improving the productivity of the quality control. Similarly, for the other like colorimetry, it is a pharmacovigilance method where the water content determination, it is by colorimetry. Can we establish equivalence and on the Carl Fisher, and can you means make the alternate to that. So similarly, means lots of techniques are there which you can look for the simplified method. Like if you see that heavy metals, earlier USP method, it was very conventional method. Now that elemental impurity has come because it was very tricky for the analysts sometimes. When they try to make that 20 ppm or 10 ppm standard, sometimes they used to get, sometimes they don't used to get that reference solution. So that's why many of the things even the pharmacopoeia looks for, and there should be appropriate justification and scientific rationale for why we are choosing that method. So with that, there are options and we certainly should look for, right? One more example to add to that we will tell. Like there were lots of discussion with that investigator, pharmacopoeia, means individual regulators regarding this USP have a different method, EP has a different method, and we perform stability study using USP method, EP method, there is some IP method. So now we need to look for that what is the best appropriate method considering the separating all impurities. And we are successful with the regulators also by filing the update, and we are doing not only for the stability, we are going for the release method also. Why we should not, right? So there are lots of opportunity where we can.
Okay, thanks, Manoranjan. Prashanth, your thoughts on improvising the current methods, and what is in the future for the new technologies that would come in the laboratory?
Good afternoon, everybody. I'll just take an example. I had some passion, went to a big hospital here called AIG. I looked at the entire work stream and the flow of samples. Just to give you an example, they manage 20,000 samples a day. Whereas we in pharma laboratories, we hardly manage 7,000 to 10,000, at least I can talk about my company, we manage about 10,000 samples a month. So how is it possible for a diagnostic or a hospital kind of a facility to manage this kind of samples? If you look at their automation, and of course I'll come to the disadvantages of that, they just pull the tray, the samples are directly kept, it has a complete turret and they put, and all the results are available. Each rooms are segregated. Okay, so how those applications can be implemented into pharmaceutical laboratories is a thought which came into my mind. When we looked at a lot of literature, okay, right from the value stream mapping as we call, right from the sample receipt to the generation of the result and the COA, what are the various unit operations which are involved, the unit operations of the laboratory in my language. Right from sampling, okay, we have highly sophisticated NIRs now. We don't even have to open, take into the sampling area. The identification is quickly done. When it comes to the sampling, after post-sampling is the sample receipt at the laboratory, where you have enough of barcoding systems, the tracking, tracing. Earlier days, we used to see a very thick register. All of us remember who are worked in the laboratory in the past. A very thick register, it could be some 200, 300 pages, and every page number will be typed, and the title of the register will be written. Every sample right from date of receipt, name of the material, pharmacopoeial grade, quantity, GRN, whatever you want to tell, you have to write by hand. Today all those things have gone, you have just a barcode, you get entire information. That's the second step of the entire flow. When it comes to the analysis, there are different techniques, as all of you are aware. If you see the latest, again it's not very popular. Some of the companies, I can tell, Agilent for example, has an instrument called TRS-100. So this is a modified version of Raman spectroscopy. It is called as transmission Raman spectroscopy. So if you take this operating principle of this instrument and correlate with the hospital example what I told, these are non-destructive tests which are done. Basically, the entire percolation of the Raman spectra into the tablet, which can differentiate the APIs and the excipients, and you can get into a non-destructive type of assay analysis as well as the content uniformity analysis. I was doing a rough math out of my curiosity. The cost, not only the cost, the time of analysis will be 1/10 of the HPLC type of analysis. Suppose HPLC needs a 24 hours to 36 hours for running one complete sequence. Here in a span of 6 to 8 hours, you can complete 10 batches on all non-destructive. If you look at the human error is one of the topics which we will be discussing. The majority, at least 50 to 60% of what we see, at least the data what I come across, the sample preparation is the biggest cause of human errors. The moment you eliminate sample preparation itself from the system using these kind of techniques, your human errors are directly 40-50% of the human errors can be reduced. So even the sample, the quantity of samples which are required for repetitions and other things, you just place a tablet or a group of tablets, you get assay, you get content uniformity. And coming back to other things which Dr. Manoranjan also told, adding to that, if you look at dissolution, which takes a lot of time, today I think Dr. Ramaswamy is also here, he's a dissolution expert. I'll be very small to tell these things. Dissolution coupled with UV, dissolution coupled with HPLC, the intrusion of a person in dilutions, again dilution is part of sample preparation, it leads to human error. So if you really see, you place a tablet, you get the result with the UV, you place a tablet into dissolution, you get a result with the HPLC. So these techniques are already available. Only thing is, we need passionate people in the industry, in my language, who can really take up these techniques and see, take it in a larger way is where we need to see. An element of risk is always there for the leadership. The cost and benefit has to be justified to the management. So the passionate people can definitely take these things forward, is my opinion. Thank you.
Thank you, Prashanth. Prashanth, you mentioned about the logbooks and the passionate people. Yes, we have got a lot of passionate people, but I can tell you that in the industry, in the companies also where you have a laboratory, and if they start automating their instruments, the softwares are able to take care of the logbook. But the irony is that the logbook still continues. Okay, so as you said, that there are passionate people, so they want to keep that passion of having the logbook, and then they say, oh, somebody made a mistake in that logbook, so there is a human error. But my dear friend, why did you keep the logbook when your software is able to take care of this? So these are some of the things that the people have not. And the answer is that the QA did not allow us to remove the logbook. So that's a very smart answer that is given. And I'm sure that we have to come out of that mindset to make sure that the analysis and the automation really takes a very positive stand. So over to, yes.
So I'd like to respond to what Rajiv just said. He said, QA says keep the logbook, right? Honestly, what I will ask you all, hand on heart, are you ready for automation? Honest to God, we all talk about automation, but are you ready? And only you can quietly answer the question. अरे बाप रे क्या हो जाएगा? ये ऐसा आएगा? अभी FDA के देखेगा? Oh my God. So sometimes we say, and that is what I said to Rajiv, we say big things. Do NIR, do Raman, do SOTAX, do this, do that. But I don't know if we are ready. And why are we not ready? We are not ready because our process is not so rugged. The method is not so rugged. So we want both. We want to toggle between Activa, that motorcycle, and Lamborghini. Can't do both. But I don't want to take your time, but he just, he said something. I had to say something. God's given me a mouth. So please, continue.
Yes, Surya, go ahead. So again, the same questions on, what can we do in the current practice and what is the new technologies that are available?
Yeah, I think, good afternoon, ladies and gentlemen. We need to look at two aspects. One is, the laboratory automation, especially, in terms of regulatory environment. A lot of challenges from US FDA especially. And hence, not only 21 CFR Part 11 compliance requirements, but laboratory automation has come into, the picture. And the second thing is, we talk about automation, new instrumentation and all. But at the same time, delivery point of view, the turnaround time. If you are going to release an analysis, which I can release in a day, somebody can release in a week. So there we are losing, a lot of productivity. So in that aspects, like a few examples, if we can switch over the HPLC to UPLC, the runtime will be very less and we can minimize the turnaround time. And similarly, the other test, for solvents, especially in the bulk analysis, we are repeating, 2 times analysis. Initially collecting the sample from the tanker, then again, recirculation, then followed by full testing. By applying, NIR methodology, which is acceptable for regulatory agencies as well. So I think we can reduce and, minimize the turnaround time. So just these are, couple of thoughts from my side. Thank you.
Thank you so much, Surya. So that brings us to our next question to Dr. Ranjana. Dr. Ranjana, we talk about all these automation, but that's for the QC. But what do you think are the benefits of becoming a factory of future? I mean, in terms of efficiency in the manufacturing and efficiency in the quality control.
So, factory of the future sounds wonderful, right? And we all want to be a person of the future, right? We're constantly re-engineering ourselves, redesigning, reimagining. These are the new buzzwords after COVID, right? Reimagine, redesign. So I think there is a lot of goodness in automation of the factory. Because as we just heard from Dr. Reddy, today the marketplace is very crowded, very competitive. We all are actually competing with each other. We are sitting here very nicely drinking coffee and listening, but we're all competing with each other. Who can get to the finish line fastest? Whose drug can be approved the fastest? Whose pricing is the best? So that's one side. The other side is there's so much price erosion, so much pricing pressure. Every country. And we must look at productivity. But while we look at productivity, we can't forget compliance. So while we are making the factory of the future, if you're only looking at productivity and you've forgotten the compliance part, then we are in a problem. And if you're only looking at compliance and not looking at productivity, then also we are a problem. Because for compliance, if I need to check something, I could have myself doing it, him checking, and then him checking him, right? I'll get good compliance. But then can I automate this? So I think there's a lot of benefit in wanting to become the factory of the future because the marketplace is very crowded and there's a lot of competition. So if we don't do it, we will not be relevant and we will not be able to sustain our business. So I think it's important, but each company is different. So each company has to think, what is best for me? Because you invest, often what happens, and I don't know if you've faced this, but I've faced it a few times. You spend 100 crores, 1,000 crores, 50,000 crores, and you go and you pitch that I'm going to do this test, or I'm going to automate, I'm going to have MES, I'm going to have electronic batch record, I'm going to do all of that. And in doing all of that, I will be able to redesign my workforce. So instead of having 10,000 people, I'll have 5,000 people. But no company has really done that. So you automate, you spend 1,000 crores, you've got your electronic batch records, and you still have your 10,000 people. So I think it's a very bold decision when we decide to become the factory of the future or the lab of the future. By the way, I'm trying that too. I want to be that too because I want to stay relevant. But you have to chalk out everything. Is it really beneficial? Are you going to get that ROI if I do it? And do you have the courage to do it? So you don't have automation plus the logbook.
Thanks, Dr. Ranjana. So I think you rightly said that definitely have to look at the options that are available. Efficiency is important, not only from the laboratory, but also look at debottlenecking the processes within the lab so that you are actually able to get the benefit out of it. So the next question is to Dr. Manoranjan. See, Manoranjan comes with a very strong analytical and QC background, very hands-on. I've seen him working in the laboratory. And now he's heading the department. For him, and I've seen that he has a go-to approach, and he is very passionate about trying to bring in new technologies. So Manoranjan, to ask you this question, I know that your hands may be itching to get onto the laboratory and to work by yourself, but now that you have a team under you, not only within one department or one location, but several locations, So what would you see in the present scenario and the future of the laboratory, which can definitely make your life easy as the head of the department?
It is very much essential, and before starting that, I would like to add where Dr. Ranjana was telling about that adoption of that new technology, right? Where the process development, analytical development, it is the source for that any technology to bring in. Later on, other thing will come. Either we can adapt, we cannot adapt, we want flexibility, we don't want flexibility. That will be later on. But first thing, if the development of the method in the process, if these two things are rugged, then the adopting the technology, it becomes very easy in the quality control or analytical laboratory. If you consider starting from the R&D itself, every R&D will have that their development. At the same time, they will have that support to the quality control, routine quality control for development, analytical method validation, any investigation when it comes, that will also be there. So now it is very becoming challenging for them because they are also coming to the scanner of FDA inspection or other agency inspection. So what kind of system they should have to segregate their sample? Now R&D sample is there, plant sample is there. How to separate that? Can they automate that? Even for the segregation of that. So there is means opportunity for them to have the adoption of the automation where that even batch number is sampling in place of having the all manual recording for assigning the AR number, they can go for the automation system, right? Generating the level from there to have the proper identification of the sample. Like Prashanth was telling that diagnostic lab, how they maintain that so many samples, why quality control analytical lab cannot maintain, right? So we need to go for that opportunity, what simplification we can do, and this means anyhow you are having the batch number, you have the AR number, analytical reference number, so you generate the documents, then there is no problem for adapting that. There is no any barrier for maintaining again separate manual record. You can go for the automation, right? Same thing for that, why we cannot have that now heat and trial method when we go for the method development, right? Adapting some buffer, acetonitrile, methanol, trying, injecting, seeing either it is coming or not coming. Why it cannot be, means automate, ask that means who are providing that application, can they work out the artificial intelligence where we can give the molecule, like ICHM7, genotoxic impurity, how it works. They give the structure, everything comes. Either it is genotoxic in nature or not. So similar kind of application, it can be adapted. We can demand, we can give the our usual requirement, which can give the what is the pK value is required, what should be the pH for this, right? What can be, means wavelength, certainly you will scan for the UV. So to get lots of information, right? So starting from that development, then coming to the quality control. Lots of things presently also we are maintaining the record, like analytical balance, right? Analytical balance, is it possible to connect directly integrated to that LIMS or HPLC, right? We want flexibility because if we want to adjust some weight, we need flexibility. If we don't want, so why we should not focus on the training program to the people who should be very good, accurate into the doing the performing the analysis in place of going for the flexibility, right? Now earlier workbook where the analytical worksheet, everything used to be manual. Now you see that now people started getting benefit who has implemented the LIMS. Now everything, your working standard cancellation, your stability sample cancellation, your schedule, right? Everything within a fraction of seconds, everything you are getting. How many number of samples is analyzed, that you get. So every information you get at a time. So that is the advantage of that. There are lots of manual process where with the checklist we review that audit trail, multiple activity we perform. Can we go for that robotic process automation with help of whichever manually people do, lots of time, 4, 5 people are per shift getting involved into that, reviewing that analytical data. For that, either it is reliable or not, because quality control is considered the mirror of the any organization. If the quality control data is reliable, means your organization is reliable, considering that. And any inspector or auditor, because all-time readiness we talk about, right? That is the most important thing which we need to look into. So can we go for the robotic process automation where it will be only help to run through that application in place of human being, one robot or two robot will run, means evaluate the total data. Either sometimes we say that manual integration should be authorized. It is not allowed, right? Now, it is not allowed, but either analyst is updating to the supervisor that I have done the manual integration. We don't know. Thousands of chromogram every day you are generating. Either that all thousands of chromogram people cannot miss out to see that. There is a possibility that they can miss out to see that. But if we go in the automation process, then everything will come out. Everything will get filtered within 5 minutes or 10 minutes, and you can review the data. Anything it is done, either in heavy integration, manual integration, whichever activity you have done in the audit, in place of going through each and every line item, seeing manually, you can go through the system will work, right? So there is that kind of opportunity where we can utilize automation and we can take lots of benefit of it.
Manoranjan, that's a good point that you mentioned. I can tell you that many times the failure or not being able to implement automation is because of the head of the department is himself not so very confident. That confidence, if it is shown by the head, by the person at the helm, I think automation and also improvement in the technologies come very fast. And I know when you have a person like Manoranjan who himself has that kind of risk-taking ability, okay, I would say, in terms of not being perturbed by the fact that risk of failure, I think this becomes a reality. And this is something that I've personally looked at this kind of issues where the youngsters are ready. The youngsters would say that we want automation, we want to do something in a much better manner than the conventional way. But the person who is heading the department, he's not ready. And that kind of barrier definitely is one thing that has to be overcome. So I think that's where I think people like Manoranjan, I've seen him with that kind of guts that he has, saying that yes, I'll be the one who takes the responsibility of failure. If it is a success, the success lies with the team, but if it's failure, I'll take it up. But he'll make sure that there's no failure. So thanks, Manoranjan, and that's the kind of ability that you have. And we want more people of this kind. Yeah. So coming back to Prashanth, Prashanth, your company has been into scaling up operations, and we have seen that the scales of operation of a company has been some of you do something which is completely out of the world compared to what the others are doing. It's basically the manufacturing. So is it something that you also looked at the analytical part of it, or is the analytical part still continuing with the same methodology, or how has the debottlenecking been done in terms of the analysis, which is so much required when the scales of operation go up to a large level?
Yeah, sorry. So just to give you a right perspective, I come from Granules, and we are one of the largest manufacturers of pharmaceutical products. On a monthly basis, just to give a right perspective, we do about 2 billion tablets per month, and we are ramping it up to 2.5 in the next 6 months in one of the largest formulation facility. The similar scale of operations when we talk about in one of the other facilities of API, we do about 2,500 tons of API per month in one product of paracetamol. So now the quality control has to gear up to answer Dr. Rajiv's question. The quality control has to gear up with this scale of operations, where the manufacturing scalability is continuously ramped up. If the laboratory is not ramped up, as Dr. Ranjana told, we are not in the competition, we are not in the race, we tend to lose out. Being in the generic space, okay, and as she rightly told, companies are competing with each other. So the pricing margin in the US is significantly decreasing. People who are into the generic space definitely do understand this kind of competitive pressure in terms of pricing. So what we say in Ramayana's Alilu Seve or a squirrel's contribution, though the cost of quality control is minimum when you compare with the entire the operational expenses of the companies, so still we can contribute significantly from the scale up. So having set this backdrop, so how do we cope up with the speed of manufacturing is one point I want to address. So there are different ways of deliberating this. The deliberation which me and my team has worked out, some members of my team are here, Mr. Babu and Rajesh. So we have worked out with a very innovative concept which we believe. What are those innovative concepts? So right from the layout of the laboratory, I'll take with one by one what we have done, which we feel has given us good benefits in terms of efficiency. When you look at a layout of the laboratory, Kivani, I think the Kivani person was here. He made a beautiful presentation on the infrastructure. The design of a laboratory plays an important activity in terms of efficiency. Many people may not believe, and this is fact. He showed some numbers on the safety index. I think he's here. Yeah, he showed some numbers on the safety index. Similarly, the layout, if you see, if you have a sample management in one place, a working standard is another place, the documentation of spec and STP is in another place, the column is in another place. When we kept a stopwatch to our hand and started calculating, there are almost about 25 minutes to 30 minutes of 8 hours time where an individual analyst will be spending his time in searching of these things. So if you look at layout, so you design a layout in such a way, the samples come to the laboratory in a particular room, the full kitting in terms of spec, STP, column, working standard, all the documentation are given as a full kit to an individual. As per the shift schedule, the planning is done. The person comes to his place, he just picks up a tray which is already ready, he goes to his workplace and starts the analysis. This definitely gives a significant impact on efficiency up to the level of 8% by number. So is there an investment? It's a zero investment. Is there additional manpower required? It is zero. Is there an automation required? It is zero. Just in terms of a layout, an 8% efficiency can be improved in a laboratory, which we have demonstrated in a span of 1 year. So this is a significant in terms of layout. Second, when it comes to the scalability, we talk about human errors continuously in our systems, in our pharmaceutical industry. So how do we minimize human errors? So the day one a person joins a company, within 3 months he wants to run a HPLC. He wants to run a LCMS. That's the ambitions of people, right? So nothing wrong in the ambitions. So if you divide the people into their scalability or the skill sets, basically, when the lab unit operations or the tests are classified into a level 1 test or a level 2 test or a level 3, level 1 being the low skill and level 3 being the high skill test. Level 1 includes in terms of a sieve analysis, it includes some kind of a bulk density apparatus or bulk density or a tap density and things like UV, things like solubility. If you classify, you don't need a person with 10 years of experience or a PhD person to do these kind of tests. A low-end level of jobs are enough for these kind of operations. When it comes to L2 tests like a dissolution or in terms of an IR, and L3 test like an LCMS or a GC coupled with MS or a HPLC. So this leaves high level of skills basically. So if you look at the whole ratio of analyst to non-analyst in a laboratory, a 70-30 is an optimum ratio of analyst to non-analyst in an ideal pharmaceutical laboratory. So in this 70%, if you divide this into level 1, level 2 and level 3, so the level 3 makes a significant portion where the skills are really required. We really need where the errors are happen more. So if you divide the laboratory based on the skills of the people, more than the skills of the people, the skills required for that unit operation or the test. So there will be a lot of efficiency improvement. There will be a lot of human error reduction is one more thought. Now coming back to the other aspects I told about the TRS, wherein the efficiency can go up. Again, it's not proven as such. There are hardly 1 or 2 instruments in the whole country, what I come to understand, but we are definitely exploring. With respect to dissolution coupled with UV, dissolution coupled with HPLC, when a rough cut calculations are done, it needs a significant investment, but as Dr. Ranjana told about the return on investment, it is worth trying it out. And we are in that exploratory phase where we are in the CapEx discussions. And last part is reduction of human errors, for example, where we are coming out with a technology. We believe, again I'll take a disclaimer, we believe that we are the first of that kind in the country. We are coming with some smart glasses for the analysts. So what are these smart glasses? These are expensive smart glasses. When a person wears that, which is connected to a particular software as well as the Wi-Fi, a person is able to see the SOP. The person is able to see the STP. Today, I'll just deviate the discussion. The SOPs are in some place, so it becomes a showpiece for, because it's a, as Dr. Rajiv told, QA asks for that, somebody puts an SOP and it's there in the folder. Now if you see this technology of smart glasses, the SOPs are displayed on the specs itself. This technology is used in Boeing, this is used in Mercedes-Benz, and this is used in all the German companies. So these technologies we have already started, the URS is finalized, the party is finalized. The SOPs are seen, the STPs are seen from the glass itself. When it comes to a calibration, when it comes to a particular critical step, if it is properly defined, the person can see the entire P&I diagram, for example. He can visualize the entire HPLC where a pump is there, where a detector is there, where a carousel or the auto sampler is there. He is able to visualize everything through a pictorial representation in the system, and he can operate the respective sophisticated instruments. This will definitely reduce the human errors significantly. And at the end of operation, he is able to take a print or save the data what he has done. If there are 10 steps on a conceptual thing, all the 10 steps as he followed, there is an evidence which can be generated after the completion of his particular test, which gets into a archival and which gets into a data retrieval which can be demonstrated to the auditor. Especially in terms of preventive maintenance, especially in terms of calibration of instruments, or even from a manufacturing perspective, the preventive maintenance of equipment. With these smart glasses, majority of the activities can be completed with this cost effective, at the same time reduction of human error, and of course, what you call human error reduction coupled with data archival and data retrieval. So there are several things which we can always deliberate, implement and do this. This journey we have started in granules, both in manufacturing and in laboratory. Thank you.
Very nice to know, sir. I think in your laboratory, you have James Bond working in the lab with all the automation which is going on. So nice to know this. Coming back to Surya. Surya, your company is mostly or primarily into custom synthesis for big pharma companies for their new products which are planned to be taken up for NDD also. And you get a lot of projects, and I know that you have been the pioneer in one of these initiative of custom synthesis. Now, when it comes to custom synthesis, the expectation is the quality of the material should be as per the requirement. The process is most of the times is given, but you need to do a lot of scale up and also do a process development. But is there something that they also talk about the analytical part, which would be a kind of a futuristic approach in terms of analysis rather than going into some conventional method of analysis, or what is the approach that they follow?
Thanks, Dr. Rajiv. I represent from Neuland Laboratories. The company basically, established as a generic player, but last 5-6 years, it is diversifying towards custom synthesis. Fortunately, I had experience of handling these customers while I worked with Dr. Reddy's as a CPS QA lead. So that with that experience, it becomes easy for me to handle these customers. We have small, medium size and big pharmas. Most of the projects are, phase 1 developmental, phase 2 developmental, and some projects are, matured towards, commercialization, so that there are NDA filings. So one of the requirement, if I assess properly, from all these perspective, most of the players are from US, and the requirement is very clear that, the documentation is most important than the material. The material quality comes after documentation. They trust and believe mostly into documentation, whether it is a batch processing documentation or analytical documentation. So for analytical, mostly they insist us, more automation and the advanced techniques. So that's the reason, our laboratory is completely automated now, except a few analyses in the wet area. And we implemented LIMS, electronic signatures, electronic logbooks. So that is the primary requirements, especially come from these customers. Otherwise, it might have taken more time also. And they also insisting us towards the operations automation. So we did not commit much, but going ahead to implement the electronic batch record is we are not daring at the moment, considering the risk involved as well as the kind of experience faced by Dr. Reddy's and Lupin. So other than that, I think digitization point of view, we are clear. I think we are going ahead, to implement some of the automation techniques in the operations as well. So this advanced techniques and automation is top priority for these players.
Thanks, Surya. So that brings me to the last question to all the panelists, and then we have some time for question answers from the audience. We talked about all this automation and the new technologies that will come up, but what do we do now? Because I know that we can spend a lot of money, we can invest a lot of things on automation and get the things done. But the biggest concern now for the industry is how to reduce manual errors, how to minimize our out of specifications. So in this regard, I would like to hear from the panel members here of what is the first thing that they would do. I mean, I know the management, top management is eager to invest, but what are the first things or the most common things that you would do to minimize the errors which can reduce any kind of inefficiency in the laboratory and the plant? And of course, the next layer is to go for automation. But in the current scenario, what would be the best practice? So Dr. Ranjana, to you.
So I think this is the heart of the question. So we all know that automation is great and we all want to automate. But you can't go tomorrow morning and start automating everything, right? Your lab or your manufacturing facilities. So how do we live? How do we continue our operation? I think it's extremely important for each one of us to know your product. Know the product in terms of, again, I cannot emphasize on the ruggedness of the product. Because if your product is not rugged, and if your method is not robust, where everybody and anybody can do it, meaning the validation is not robust, no matter what you do, errors will come. And unfortunately, those errors sometimes get misappropriated, and it becomes a human error when it's not really a human error. It's the easiest thing for most of the industry to say it's a human error. But what my request is, go back and check, is it human error or is it a manual error? But what was it caused from? Was it caused because you couldn't separate the peaks? You got a shoulder? Or was it because the product itself is not homogeneous? So you get one tablet 20% and the rest 100%. And that also is lumped into human error. So I think, Rajiv, it's very important for our industry and all of us to understand is you want to automate, you should automate, because I said you must be productive. Otherwise, you're not going to be relevant. But while you're doing that, while you're investing in the MES, LIMS, and digitization, and all of those wonderful things that are available, let's go back to the basics and have a good monitoring system. Have your procedures written in simple language where everybody can follow. I think that is not done often. Have an SOP that is 10 pages. How many pages is your SOP? Average? Anyone bold enough to raise your hand and say, I have 100 pages SOP? Yes, sir. 40 pages. He says 40 pages. Anybody with 80 page SOP? Don't be shy. 80 pages. Anybody with 100 pages? I don't care if it has annexures. If you have annexures, that means you have not written it properly. Why can't you embed it in? Because that SOP will never be followed. So what I'm saying is, to your question, if we can't automate, have good procedures, ensure people know. We talked about augmented reality. That costs a lot of money, you know, with those lenses, and you start doing all of that fancy stuff. But most companies don't have that. So how do you live? Have someone monitoring, because unfortunately most of the supervisors are preparing PowerPoint presentation. They're not in the lab, they're not in the factory, they're busy preparing PowerPoint presentation. And then something happens, there's no root cause, because root cause you go and find after 30 days, there's no root cause. Sorry, I'm being very open and honest because you all understand. I see some of you smiling. You know, that's what's happening in your factory. So what we have to do is we have to stop that. How do we stop that? Go back to the basics. My friend, there is no substitute than the basic. Have simple procedures, have simple SOPs. If the language is not understood, English is not our language. If Telugu is the language, put it in Telugu. You go to Japan, everything is in Japanese. Why do we insist that everything has to be in English? Have a translator. I know some of you are saying, what the hell is she saying? I mean it. When you go to Europe, it's in German, Spanish, French, Italian. So when our people cannot understand the language and we expect them to do something, it's not possible. So basics, easy procedures, easy SOPs, easy test methods, and your mantra should be, your SOP should not be more than 10 pages. Nobody is even admitting. See, they're like, what is she talking about? Yes, sir, you agree with me, right? Mr. Sotax, you agree? See, I get this. He is the guru. He's the guru. He's a very senior person. So that is what I would say. You know, stick to the basics, and then we can dream about super automation, digitization. Another point I want to say, when you are ready to do digitalization, which is different from digitization, one request, don't take your manual process and convert it into a digitalized process. What we do manually, that is what we want to do if we use automation or if we want to digitalize it. Don't do that. Think different. Think out of the box. And in the CFR, hardly there are 11 places where it says, if I'm the checker, I must have a reviewer. So there are many things that does not require reviewer. So when you're digitalizing, think of that as well. That would be my humble request to all of us. Thank you.
Yes, Manoranjan, quickly, because I want to go through all of them, and then we have some time for the question answers.
One thing in extension to Dr. Ranjana, regarding this, when we make any documents, when we want to go in the manual, in place of automation, designing of the documents is very important. Either it is a procedure, either it is a checklist, right? We make lengthy checklist, but either the checklist is sufficient, adequate to detect the problem, detect the issue, right? So designing of the documents is very important. If you design the documents properly, then it will be helpful for you. And when you are moving from that manual to automation, I have seen the people, they're very rigid, whichever in the documents presently in the manual, they want to go replica to that. And we should be flexible for that. So when we are fixing the user requirement, and many of the automation got failed because of that they want to implement manual system to the automation. And because of that, user requirement is not very well defined, and because of that, automation get failed. And they are totally getting locked down. So they are not in a position to go here, we should scrap that automation or what to do. So it is very important that when we are designing that any process, it should be well written, well discussed, right, as a group, considering your requirement, considering your mispractices. When we do, considering that, then there will not be any problem. one person, some senior person should sit and they should make the total documents and make that. It is not correct. There should be group of the people who are really involved into that. Another thing which we would like to add regarding the, we talk about the procedure. What about the training part? Badly, whichever we are missing into the industry on the training, focus on the training. If ERCH Q7 doesn't talk or CFR doesn't talk about the requirement of the training, I think many of the organizations, they will not make that record, they will not follow the training, right? But that is the key. If we give the proper training to the means actual training, live training, then whichever Prashanth was talking about the human error. Human error is a source of lack of training. We don't give the live training. We tell that you read the SOP. Even the people, when they explain the training, they read the SOP, total SOP, 45 minutes, 1 hour, they read the SOP. Then it will not be effective training. So the training is also important for making the documents and giving that demonstration. Thank you.
Very good point, Manoranjan. Yeah, quickly, Prashanth, on what are the things that we should do to minimize errors, especially human errors, without even looking at very hi-fi automation?
Yeah, absolutely. See, automation is one thing. I talk about an iPhone. I hardly use very few features, but it has so many features. If my son talks about that, he knows many of the things more than me. But we talk about automation. We also spoke something about fully automated but manually operated. So this is called a FAMO principle. Like our NAMO, it's a FAMO principle. So having said that, what is that we cannot do with the basics? Like as Dr. Ranjana told, what are those basics? If the fundamentals are strong or the basics are strong, we can build upon. So once the basics are set right in terms of a training, in terms of an education, in terms of skill development. She also mentioned about a PPT presentation and people spending in PPT presentation. I'll just take 30 seconds to tell. When I was a kid, maybe 5, 6 years, I still remember we used to go to a family doctor. And that doctor used to spend a lot of time with us actually. In the sense, when I go with my mother, my father, he used to spend a lot of time. He used to hit us. I think I spoke to Dr. Ramaswamy some time back on this. He used to hit, he used to talk for more than half an hour. He used to give one tablet, next day we are ready, whether it is my father or my mother or me. Today, doctor, when we get admitted to a hospital, if we get 3 seconds or 10 seconds or 3 minutes, rather not seconds, 3 minutes, we are the luckiest people in the entire day. So having brought this quote, if you look at the time spent on the newcomers in the company, it's extremely important. The day one of a person who joins, either a fresher or an experienced person, some 100 SOPs are thrown on him, and next 1 month nobody is talking to him also. So this is, as he rightly told about the training, CFR, ICH, which Dr. Manoranjan told, we leave the people, that means we put into a ocean and tell him to learn swimming. That is our pharmaceutical industry today, to the large extent. I may be little rude, but this is reality of the industry. So if the time spent, like the doctor's example, you spend more time with the individuals. If the sectional heads or the managers or the senior managers spend more time with the people in making the basics understand and develop them as a leaders, develop them as a potential for the next 10 to 15 years of this industry, I think we will be winning the battle in that front, is my opinion. Once we are saturated with all these manual things, then comes the requirement of automation in the interest of efficiency, is my belief. Thank you.
Thanks, Dr. Prashanth. Of course, if you got cured with just one tablet in those days, there were 2 reasons. One is that you had a fear of those bitter tablets, and the second is that you had the will to get cured.
It was granules paracetamol.
Okay, Surya, your thoughts about what are you doing currently to make your system more efficient in terms of in the current practices, and what are the areas that you look at to reduce manual errors?
Thank you. doctor, we, as a company, what we are doing is we bifurcated the errors into three categories in the laboratory especially. One is, man-related, that is analyst errors. And the second one is method-related. And the third one is instrument-related. So always the man-related is more contributing, more than 50% in all three laboratories. We have 3 units and 3 laboratories. So I strongly believe that the kind of steps we have taken is the robustness of method. While coming from R&D, once it is developed and transferred to the site, whether this method is really capable enough and to get reproducibility. So method transfers, everybody is doing method transfers, but those method transfers as adequately we are doing is important. Where the giant analysis from analytical R&D and the quality control with protocol-based approach should not be superficial. So that should be adequate enough. And the second thing is the procedure. Once the method is transferred to the site, whether the simplification, as Dr. Ranjana rightly mentioned, that it should be easy to the operator, understandable, and it should not become heavy. And the third criteria where we were looking is the instruments. Most of the instruments are some of the old laboratories, if you look at more than 10 years, then you may not expect the reproducibility as well as accuracy, and there will be lot of breakdowns. So what we did as a company, 8 to 10 years, more than 10 years, we are not going to continue the instruments. So we are going to replace from 8 years to 10 years. So that is one. And the last aspect is the training. The training, if you call all the analysts and sit in a classroom and provide the training, that is not going to be much beneficial, what I believe. If you go with the method to the HPLC or GC or wet analysis, stand up and maybe 20 minutes training also effective. So by doing this, I think a year back, our analytical errors were almost 40 to 45%. Now it has come down to 10%. That means if a factory is having 10 OIS, minimum 4 to 5 OIS just because of lab errors. Now it is 1 lab OIS out of 10. So that we achieve.
Thank you so much. I think what I take from all this conversation is Dr. Ranjana spoke about the basics, about reducing the number of SOPs, being very smart. Manoranjan and Surya touched about the training part, and Prashanth also talked about the way that your work should be done in a smarter manner. So thank you so much. We have a few some time for some questions from the audience, and yes, please.
Good evening, I am Sudarshan. It was really a fascinating session. While we can see the experience and expertise sitting on the other side, I believe we need to take the outcomes and experiment upon to evaluate the expertise. Having said that, morning was a session focused on science, and afternoon was a session focused on fusion between science and technology. It was absolutely fantastic. Well, I would like to ask a specific question to all the panelists with reference to an emerging technology, bringing in emerging technology, as to how can we make use of this emerging technology in interpreting, which will play a very vital role to minimize the errors and enhance the productivity. Like, if you have a solution for interpreting the bathochromic shift, interpreting the hypsochromic shift, interpreting the hyperchromic shift, interpreting the chemical shift, interpreting Jablonski diagrams, will it help you to enhance the productivity in the lab, wherein the errors are minimized to the maximum extent?
Anybody wants to add here? Yeah.
With respect to minimizing the errors, it will be the combination of that automation as well as the training. Because automation can bring that many of the things which we are thinking, which presently we are visualizing, that either it is possible. Payment, is it possible? Right? Through machine. Either dilution, it is possible. Like dissolution, it is possible. Connecting with the HPLC. Maybe for the payment, it may not be possible at present. So again, the training is required. How to take accurate payment. Again, transfer. Again, dilution. Right? So again, the training is coming into picture. Either live training, if we give to the analyst, individual analyst, then we can achieve that minimizing the error. The source of the error and the confidence of the analyst, it comes from there. Because that analyst is not confident about that what is the payment they are doing, is it really accurately transferred or not transferred, or something has been spread, right? So what kind of technique, either through that butter paper to transfer to the volumetric flask, or directly keeping the volumetric flask, giving the exact transfer like solvent we are transferring, right? So the training plays very important role, right? Something whichever with respect to reliability of the data, where the automation will come into picture, it will help to minimize the error. Like when we say about the robotic process automation, manually when we have the checklist, we verify that all checklists. It is becoming kind of traffic police job. Every day means raising the hand, stopping, that kind of thing. So people are habituated to seeing that checklist. They have the habit for making the tick mark. When we go to the automation, it helps that machine do. Human thing, lethargic approach at that time, that individual mindset, what is that, either he's disturbed, he's in jolly mode, or what kind of thing, it will not come into picture. So it is a combination of means training and automation. It can help to reduce the error significantly, right? But considering that present scenario of pharmaceutical industry in India, where the lots of company has not gone into the automation, right? Many places, LIMS has gone, but robotic process automation, people have started getting benefit of that. Can we reduce the number of manpower, resource, right, time to increase the productivity? So people have started going where the repetitive work is there. Can we go with that automation? Where that science is there, where the accuracy is required, training is required. So that is a combination. I hope that I could be able to answer your question.
Hello. Hello.
Yes,
Can you hear
doctor.
me?
Yeah.
Yeah, I have one question. It's good. I think we said that in warehouse we have automation, barcode readers are being used. Our product doesn't go to only advanced countries. It goes to domestic also in some of the less regulated market, I mean to say, which are not that advanced. And I have seen that many times, especially the Indian FDA, when they come for inspection, they don't know what is barcoding. When they enter the warehouse, they only know what is yellow label, what is green label, what is red label. How we are going to overcome those issues, because for them, their standard is when material comes, you have to identify which from the distances can be visible, that this is in this status, this is in this status. You do automation, but for Indian FDA, you have to keep another standards to comply with that. How you are going to deal with such situation when we talk of automation and barcoding and this?
Let me answer this. Most important is that we realize that the inspectors who come from our Indian regulatory agencies, there's a very big initiative being taken to train them. And good that we have Dr. Rajiv Raghuvanshi, who is now the DCGI, and he comes from the industry background. So I can tell you the interactions that we have with him and the way that he has tried to make sure that his inspectors are updated with the latest technology is something quite unique. And as you know, during the inspections also, the regulatory inspections from USA or any other country, it's mandatory that they inform the CDSCO and DCGI about the inspection. So at least one person is there. And by and large, the inspectors who come and watch the inspections are really keen to know and understand and learn what are the new things that are to be done. And the new breed, I would say, the new breed of inspectors are really hands-on, are very advanced in terms of their understanding. And I'm sure that they will be updated with all the new technologies that we implement and will not be struggling to make them aware of what we are doing. And gone are the days that they will look at the ropes, the color of the ropes or the color of the labels, and they will understand the barcoding and the QR codes also.
Doctor, can I?
So can I just add to what Rajiv just said? So I think we're sitting in India, and we're proud to be sitting in India. And I request our companies that are being represented here to open their doors for the Indian investigators, inspectors to come and learn. I think it's important. The industry also has that responsibility. Now, switching to highly regulated, US FDA. There, not all the investigators understand electronic batch records. I swear, recent example. All inspectors do not understand review by exception, which is what we do at Dr. Reddy's. It's a beautiful thing. It's a bot that goes and does all this review, and you do review by exception. And it took us so long to explain to the investigators that this is how it's done. So I guess I'm rooting for our Indian investigators. Open the doors, let's train them, and let's make them at par. Because what will happen is if they become at par, our industry, I'm talking about Indian industry, the baseline will increase. And I think we owe it to ourselves. So we make better product, better quality of medicine, so we continue to just get better with one tablet.
Doctor, just for 30 seconds.
And we
30
don't
second-
only restrict to red label.
Yeah. Doctor, 30 seconds to answer what Dr. Haldankar sir has said. Technology has advanced in such a way, like say for example, if you take an S4HANA system and you have a barcode into any of the container, you put a scanner onto the barcode, it will flash green light. It will flash yellow light. It will flash red light, giving the status of the batch. So I believe that the Indian FDA inspectors will be always happy looking to those lights.
Sudarshan, that's a very good idea because people are more used to visual things rather than display. So if we can come out with these ideas, I think that's better. Yeah. Yes, Dr. Ramaswamy.
Hello, my name is Dr. Ramaswamy. I am the Managing Director of Sotex India. First of all, congratulations. It's a relevant topic for today's and future. I have been experienced with automation of lab automation for the last 12 years. Very small period only with me. In that I learned how automation can be successful. More than that, how automations are becoming failure. So I have a solution for those who fail to become successful. And we have two gentlemen sitting each other who has been very successful with automation. And I am part of this. The first person who introduced automation in dissolution is none other than B.M. Rao. B.M. Rao can definitely speak. He did 10 years back fully automatic robotic dissolution successful, and he took a major decision of going for automation. Lot of challenges were there. We supported. One main reason is they didn't go for one machine. In a small lab, they took for 3 machines, told us to always should work 367, 24/7. And we made it happen. Our engineers worked night repairing or preventive maintenance of one machine. 2 will be, and you know how many products worked on that machine? Just 1 NCE. 1 NCE, only stability testing for 4 years. So you must all ask anyone before going for automation, take him as a consultant and ask him, he can make you successful. The second person is sitting here, opposite side, none other than Rajiv Desai, who made automation successful in Lupin, not in one unit. And the third person is working hard, and one of the early, automation specialists is no one other than Dr. Ranjana. And she is representing today one of the lighthouse, where out of 2 lighthouse in India today, which we all will witness 20 lighthouse in the next 10 years. So we will work together and make automation successful. One example I will give you. We keep double standard in India. When it's company, one standard. When it's to ourselves, another standard. I'm sorry to say that Every year, you all buy mobiles. Once in 3 years, you change mobile. And we heard that once in 10 years, he will change the instrument in the company. Correct? When you change the mobile, do you go back? Last year 30,000, this year 20,000 or 60,000? Last year 60,000, I go as 1 lakh rupees or 2 lakh rupees. So why not this type of budget increasing for the purchase of companies' instruments? Is it happening? No. Commercial managers, what they want to come to and tell MD, I purchased 2 crores, next year I got it negotiated with 1.8, then 1.7. So commercial will get more incentives for reducing the cost of investment. And who are the sufferers? We are the sufferers. Are we talking to the management this? So these are many questions I can ask near. Because want of time, I'm not speaking much, but it's a very relevant topic. Thanks to the organizers for bringing up. Please bring it every year, and next year I will also join the panel and speak more about automation. Thank you very much. And my last request, we have brought 2 or 3 fully automated robotic instruments to this exhibition. At least all of you please visit, whether you want to buy or not to buy, at least see the instrument, which may be 1 crore, 2 crore, 3 crore. Have a look at the touch and feel it. In 10 seconds, you will get the live demo at Sotak stall. You are all welcome there. I'll be there. Thank you.
He's correct. Yes, Deepak.
I think, can you hear me? What he has said and actual situation in the industry, I think all companies are looking for cost saving. So every time they will bargain, whether it's API manufacturer, this to reduce, not to increase. So anything you get automated thing, it will be always an increased cost. That is the one thing we should understand. Second thing, I think Surya has said very relevant, that our big problem is analytical development. We first develop our products for particular market, say US, and when that product pick up, we want to extend that market to European, Pharmacovigilance, other countries, and where the methods become different, requirement become different at time. We don't plan at the beginning only that this product is going to go like that. And that is where Ranjana's question was there, that we should make 10-page SOP. It doesn't remain 10-page, it becomes 40-page. And making that 40-page, the QC guys have to work so hard to demonstrate the validation of VP method, BP method, whatnot method, every method they are the body, and that's how the SOP become 40 pages. Am I correct or not? So this is how the changes happen, which is, I can say, unplanned changes, because your product has picked up. Management now want to make use of that. So I think Surya has said very rightly that anything, if it is properly validated as per your requirement, the workload when you transfer the method to QC and other labs, multi-locational lab, it's a big headache. The verifications, all those type of things can be avoided. I think that is what I think, that if we reduce at the beginning only the things, it will be less load for the QC and no changes can happen.
So I think we have spent a lot of time now. So thank you so much, Dr. Ranjana, Dr. Manoranjan, Prashanth, and Surya, for your insight. I'm sure that each one of you gave all your experience out here, and I'm sure that it was quite relevant for the audience here. So thanks all of you for your questions and your responses. Dr. One more question? Okay.
No, there is no question actually. Just excellent discussion. Can everybody can give a big round of applause to the panelists?
Thanks for doing my job, sir.
And I'm not doing your job. I'm going to talk something else. And I think very, very important is the automation. Because as Dr. Ramaswamy said, I implemented automation in Johnson & Johnson in 2008. Okay, very long back, with fully automated resolution. It was quite successful. And today I think very, very important is, the mindset, Because today, honestly, you tell me, UPLC, what is the success of UPLC? Very less, because R&D is not interested to implement UPLC. Still all our methods are 80 minutes, 90 minutes, 100 minutes. This is the sad part of the story, right? we can do, because even the pharmacopoeial method still, pharmacopoeial people are here, I request them to come with the, faster methods, Still we have 120 minutes, 100 minutes, crazy methods.
Ask them to phase out the conventional methods.
Yeah, they have phased out titration methods successfully. Earlier we have a lot of titration, now everything is HPLC. Now they have to go to UPLC.
Phase out the normal HPLC regular, so UPLC becomes the standard.
See, we are trying to eliminate HPLC, UPLC, and use only NIR.
Yeah.
So we're trying to go ahead. Only NIR, all HPLC eliminate.
And I am confident automation will work very well. Our mindsets has to change. And last point is, Madam said very clearly, procedure versus practices. Today the biggest challenge is something in the procedure, practice is different. This is the biggest gap in the industry. So we need to have a simple procedures, because people will write very complicated procedures. People think that if I write complicated procedures, my job is secured. So sorry to say this, because people write very, very complex procedures. We have to write a simple procedures to make. Okay, thanks once again. Thank you.
All right.
1 second. The last point I want to say to the leaders, not to you. One fear here is everyone who wanted to suggest automation. My practical example in the last 8 years, people are very afraid that will I lose my job if my automation does not succeed. And I have 4 examples with me who has lost job. Okay? And yes, it's a fact here. Some of them are here also. Lost job means they change the company. They know that they are going to lose job. They will go to a next company with a higher position and higher pay, because they were not able to make successful automation, not because of the machine or training, lack of support from sometime from the top management. So my request is that to the top managers, all the leaders, please support youngsters. If you want good youngsters from NIPER and other great institute to come and work within our companies, then we have to, even if failure happens, if they are admitting the failure, correct them. I just share one more example. The students who are coming in abroad, they see, I was in Semmelweis University last week in Budapest. In the college lab, they have automation instruments. They are not working on manual instruments and coming to the industry. Imagine the situation. And we are going to work with NIPERS yesterday throughout all the directors. They have 700 crores. You will see in the next 2 years, NIPERS will have automated instruments, whether your company or not having. Because the minister said, India is going to be, India is already the pharmacy of the world. They are going to make India as the lab of the world. So all NIPER directors have agreed, we will go for automation instruments only, for which 700 crores has been allotted only for this year. Yes, 700 crores. Yesterday this was announced by the minister. So I'm working very closely with all the directors, see that how the lab has been fully automated. So those students who are coming from NIPER with all automation instrument, if they come and see you in your company, manual and semi-automatic, how will they work? So think about this, what is going to happen in the next 5 years. And please ask your commercial and managing director to invest money in automation. Whether you like it or not, we need it. We will go for automation and we will make successful. And I will be with you for the next 5 years to make automation successful in this country.
All the best, sir. Come on, give it for him. Well, that was a very interesting panel discussion. Even though we had 4 panelists over here, the audience were enthusiastic to be a part of the panel discussion. But having said that, I would like to thank our panelists and our moderators for that informative and interactive session. A huge round of applause to them. I think there could have been no better end to this day's panel discussion than how it was. Quite animated, agitated, and wonderful and interactive. All right, so ladies and gentlemen, with this we come to an end of this wonderful panel discussion. I could see so many, people all glued to their chairs for 1.5 hours without moving. So it does depict that the session was quite engaging and informative. On that note, we would definitely want to thank our moderator and our panelists for being over here, taking your precious time out and being a part of this wonderful panel discussion.